Good morning, everyone. My name is Nastasia. I am product specialist from Apical Scientific in charge for UiTM Puncak Alam. And today I will be your host for these exciting sessions. On behalf of Apical Scientific, Nacalai Test, UiTM Faculty of Pharmacy and the Tissue Culture Unit, I would like to warmly welcome all of you to today's webinar titled Grow It Right, Culture, Care and Cytotoxicity in Cell Studies. Before we move to our speaker sessions, allow me to briefly introduce Nacalai Test and Apical Scientific. Nacalai Test, founded in Kyoto, Japan in 1846, which is one of the world's most established life science companies, and with nearly 2 centuries of expertise, they supply top quality research reagents, cell culture products, and also laboratory tools to scientists globally, supporting advancements in biotechnology, molecular biology, and also cell research. Here in Malaysia, Apical Scientific, which formerly known as First Base and Exon Scientific, serve as the trusted partner and sole distributor of Nacalai Test. Together, Nacalai Test and Apical Scientific are dedicated not only to supplying product but also to knowledge sharing through scientific workshop and webinar like this one. Before we begin, here are a few quick notes. First, please keep your microphones mute during the presentations. Second, if you have questions, feel free to type them in the chat. If you like to speak, kindly raise your hand and unmute yourself when called upon. There will be a short quiz partway through the sessions. And lastly, please stay with us until the end for a chance to win a Watson Biolab Special Edition Micropipette Frank Muller editions in our lucky draw, which one lucky participant will be selected. Now, without further ado, let us officially start the program. It is my pleasure to invite Professor Dr. Mizatun Hazizul Hassan, Coordinator of the Tissue Culture Unit, Faculty of Pharmacy UiTM, to deliver the welcome remarks and introduction. Professor, the floor is yours.
Thank you, Tasya. Hi, everyone. Assalamualaikum warahmatullahi wabarakatuh. Now, I'm Miza here. I'm basically representative of the Tissue Culture Unit. Okay, so I will welcome you to the faculty of UiTM for our Tissue Culture Unit, where we provide a sterile, well-equipped space for cultivating cells, and we support hands-on learning, drug testing, and research in cancer pharmacology and biotechnology, where we hope we can bridge the classroom knowledge to the real-world innovation. So let us go through some introduction about our unit. Next, please. Okay, basically we were a fully established tissue culture unit in 2009, so it's almost more than 15 years. So as we know, the Faculty of Pharmacy is committed to research, education, and healthcare innovation, and we drive excellence through the global partnerships. That's why we actually, we welcome collaborations and also to use our services. Okay, next. So these are our research. These are actually our research services units. We have the imaging unit, we have lab farm, which we house rodents, and then we have the analytical unit. We have a few HPLC systems, and we have the histology unit, and also our what you see our tissue culture unit. Okay, where we have quick facilities that we can actually help you to actually move forward with your research. Okay, next. Okay, this is our team. And we see we have these two gorgeous ladies, Puan Fatia and Puan Farhana. They are the backbone of this tissue culture unit. Unfortunately, we have to see Madam Fatia go because she is actually embarking into a very fruitful journey after this. Okay, next. Okay, what we have in our facility, basically these are the things that we have in our facility. We have our basic tissue culture unit. We have the BSCs, we have the inverted microscope, we have the microplate reader, and so on. The ones that we are proud also to have is also our flow cytometry system. We have two units. one is the Fraxverse and one is the Frax Caliber. And obviously, we have also the CO2 incubator. What is a tissue culture unit with a CO2 incubator, right? Okay, next. These past few years, we have done many activities. We have every year we have final year project induction course where we actually introduce the students the basic techniques for the tissue culture, and then we have some seminar webinars, and we try to have a yearly basic cell culture workshop, and then we do have involved with other companies to do some cell culture roadshow, flow cytometry workshop, and also one-to-one group training where we have at least small cluster of groups to come and have a workshop with us. So it is actually how to say um tailored to them so that they can have a one-to-one personal coaching with us. Okay, next. Okay, this is some pictures on our final year project induction courses. Okay, next. Master the cell culture workshop also that we have. We don't only give talks. We do have an hands-on session, not just a demonstration, but a hands-on where you have the experience to do the cell culture work on yourself. Okay, next. And this is where we are involved in the cell culture roadshow together with other companies. And hopefully with Ethical Scientific, it will be a long collaboration. So next. And this is our basic flow cytometry workshop. We have the experts coming to our facility to actually show and demonstrate and also troubleshoot. Okay, next. This is one of our uh, frequent customer. He's been coming here for more than three times, I think, for a one-to-one training. He's from Algeria. Okay, next. Okay, that's why this is the highlight of the seminar. Actually, I won't take away the limelight. So basically, we're going to have this talk by Miss Mana, who's going to talk about what is cell culture care, what is cytotoxicity, and hope it will be very beneficial for all. I know because she's the expert. So by having this seminar, we hope that this can bridge the knowledge from Japan to Malaysia, and this will gain insight. You all will have the insights on the cytotoxicity testing and cell culture care. Furthermore, we have basic knowledge and very important knowledge on the basal media. And this will be a starting point for us to build networking and collaboration opportunities for us and also for Epical Scientific and Nacalai tests. Okay, next. Okay, why do we actually do this cell culture work? Cell culture is a research that bridges in vitro to in vivo research. And it's not actually a substitute for animal, but it's actually very important for us to have the fundamental insights to guide into the in vivo implementation. So it is very important because it is a controlled environment. So we study cells in isolation, free from immune or tissue interference, and we use this for drug testing. So in terms of development of new drugs, this is one way for us to go. And then we can actually explore the mechanism, the mechanistic insight in terms of if you're going into the development and the fundamental research. This is one way. And then it is actually for a targeted approach, personalized medicine. And ultimately is for functional assays where we can perform migration, DNA damage, and also for apoptotic testing. Next. So, however, whatever and anything good, there is always challenges. So basically, in cell culture research, there is also challenges where we have Cell culture lacks full complexity because behaves differently from human body. And then the most important and very big challenge is actually the contamination risk, where this is where we require strict sterile technique. And that's where maybe Miss Mana could actually throw us some light. And then because of the cost and resources, many media reagents and equipment are quite expensive, and we hope with Nacalai Tax and also Apical Scientific, they can help us in terms of giving us premium products at a very cheaper price. And then one thing about cell culture is the genetic drift, because sometimes long-term cultures may lose original tumor traits. That's why it's important before you start any cell culture work, you have to ensure the morphology of the cells that you're using. And then lastly is the limited microenvironment, because these cell culture actually are missing some immune, stromal, or vascular interactions. Okay, next. Okay, go back a bit, one slide, please. So basically, the last tip for me is actually when you before you start your cell culture work, there are seven critical steps that you must take into account. Okay, first of all, before you start your cell culture work, you need to understand your cells, what cells to use, and basically you need to do number one, understand your passage number. So some would say that your passage number starts when your cell arrives to your facility, but sometimes some researchers would prefer the passage starts from where for example, ATCC is delivering you the cells. If it's number 20, passage 21 from ATCC, that's the passage number that you enter. But some researchers would prefer once it arrives to the facility, that's passage 1. Okay, next is the cell seeding density. Besides that, you need to understand the confluence percentage. Number 4, viability percentage, where you need to make sure that the cells are viable. In terms of confluence percentage, the best healthy cells that you need to know the confluency would be about 80 to 95%, because sometimes over confluency would risk the how to say deprivation of the nutrients for the cells. And then if this confluency is very low, could actually induce stress to the cells. And then number 5, make sure that a microplasm testing frequency done. If you have a lot of money, please do it frequently. Number 6, basically you need to understand the pH range of the culture media. If it's acidic, obviously there's contamination or sometimes it's because of overconfluency of the cells. And if alkaline, it's basically there's low supply of carbon dioxide in your incubator. And lastly, the endotoxin levels. As you know, some endotoxin might be because of the presence of lipopolysaccharide, which is partly the tissues, the wall of the bacteria. So there might be any bacterial contamination in your media. Okay, that's the tips for me. Okay, so to the next slide. So again, I would like to thank you, Apical Scientific and Nacalai Test, for actually having us to be in this webinar. And thank you so much. I hope that everyone who is joining the seminar will enjoy. So sit back, relax, and enjoy Miss Mana, because she is the expert for this, and hope we could gain more after this. So thank you so much, everyone.
Thank you very much, Professor Dr. Mizatun, for the wonderful introductions and for setting the tone for today's sessions. Now, we are honored to have with us Ms. Mana Shimosuma, Assistant Manager of the International Business Department at Nacalai Test, Japan. Ms. Mana has extensive experience in cell culture, cytotoxicity studies, and international scientific collaborations. Today, she will be sharing with us best practice on how to grow it right from culture and care to cytotoxicity in cell studies. Ladies and gentlemen, please join me in welcoming Ms. Mana Shimosuma.
Miss Mana, I think you mute yourself. Maybe you can unmute yourself first.
Thank you for waiting. Can I share my slide? Is it okay?
We can see your slide.
Okay,
Yes.
thank you. And also thank you for introducing me, Taslia. Hi everyone. Welcome to Apical and Nacalai Test webinar. I'm so pleasure to talk with you guys. First, let me introduce myself. Once again, my name is Mana Shimotsuma. Now I'm in Kyoto, Japan. I belong to International Business Department at Nacalai Test. I was previously in the R&D department, then spent 3 years in marketing in Japan before transferring to my current department. I'm in charge of developing products for overseas markets, and I also provide technical service such as planning webinar and workshop. And this is a picture of my daily working time in Japan. I usually do weight experiments. By the way, I bought the t-shirts during my visit last year, and they also actually now I'm wearing these t-shirts. I really like it, especially the word love. And here are some of my favorite Malaysian foods: nasi lemak, pandang pao, and kaya toast. There are a lot of tasty food in Malaysia, so every time I visit Malaysia, I eat too much. Additionally, let me introduce our company, Nacalai Desk. Nacalai Desk was founded in 1846 in Kyoto, Japan. We have a long history. Now we have 16 sales offices, 1 laboratory, 1 factory, and 1 logistics center in Japan. Our product line are by region, such as the cell culture media or region for SDS page with blotting and immunohistochemistry. About the HPLC columns, our brand name is Cosmoshield. 5 years ago, we celebrated the 40th anniversary. And fine chemicals and research chemicals. By the way, he is our mascot, Bottle Task. He is inspired by a Nacalai bottle, that is, and he shares Kyoto's famous site on social media while also working alongside us. During today's workshop, he sometimes will appear, so please check him. Not only in Japan, we sell our products all over the world, especially in Southeast Asia. We provide our products through Apico. Apico and Nacalai Test have been working together for a long time. I'm really pleased to talk with you guys today. Thank you for listening about the introduction of me and our company. Let's move to today's webinar. Here is today's agenda. We will cover a wide range of basic knowledge of cell culture. Today's webinar will be quite long because it will be takes 2 hours. So please relax and enjoy the session. I hope you find it valuable. Okay, so we will start first the topics: introduction of cell culture, techniques, cell type. Roughly speaking, we can classify cell type into 2 types: primary cell and cell line. Primary cells are isolated directly from human or animal tissue. On the other hand, cell lines have been continually passage over a long period of time. The difference points between primary cells and cell lines are, in brief, cell lines are easy to culture or maintain. On the other hand, primary cells are difficult. For example, in primary cells, special techniques for isolating cells from organisms are needed. And for maintaining Ordinarily, additional nutrients not included in critical media are needed. In other words, for primary cells, you need special techniques and special media. Moreover, primary cells have limited lifespan. But primary cells are representative of in vivo tissue compared to cell line. Cell lines have lost the true characteristic of the original tissue pathway. About the way to get these cells in cell line, you can buy them from the supplier such as ATCC. In some cases, you can buy primary cells from the cell supplier, but in most cases, you have to isolate them from tissue by yourself. Recently, iPS cells have been popular. iPS cells can differentiate into many different cell types found in the body. This property makes them valuable for studying development, modeling diseases, and potentially for regenerative medicine. As I mentioned, you can purchase cell line and in some cases even primary cultured cells from commercial providers. Globally, ATCC and ECACC are well-known examples. By the way, Nacalai Desk doesn't sell cells. These providers supply cells along with a dataset. The dataset usually includes information about the cell's origin, how to recover and maintain the cells, and the details about the recommended culture medium and feeding medium. Here is an example dataset for MCF7. That is one of example from ATCC. I recommended checking at least the characteristic and the handling information section. They are very informative. Okay? Are you okay? Okay. So I proceed. In the characteristic section, you can find the details such as the origin here. For example, this line was derived from a 69 years old white female. You can also see information about the karyotype here. The model number is listed as 82, but since these are cancer cells, the actual chromosome number can vary from 66 to 87. You also find the information on expression markers here. In the case of MCF7 cells, the estrogen receptor is an important marker. In the handling information section, you will find the instructions on the recommended culture medium here. For example, MEM supplemented with insulin and 10% FBS for MCF7. It also explained how to maintain the cells. For instance, the subcultivation ratio is usually between from 1 to 3 and 1 to 6. And the medium should be renewed 2 to 3 times per week. Of course, it's not necessary to follow these instructions exactly. It's common for labs to adapt protocol based on their own practices or the availability of reagents. But I would say that dataset is especially helpful when you are working with a new cell line. Please make sure to consult it at least once. And just as a personal note, I really enjoy reading cell dataset. To me, they are almost like novels. Sometimes during lunch, I start reading a dataset, and before I notice, my entire break is already over. And we move to next. There are different types of cell culture formats. I will talking about formats. Cells that stick to the bottom of culture vessel are called adherent cells. Famous examples include HeLa and HEK293 cells. On the other hand, cells that float in the cell culture medium are called suspension cells, like HL60 or JAGAT. HEK293 cells are naturally adherent, but suspension culture is often easier and more efficient. So some cell lines like HEK293F have been changed from adherent to suspension type. F means floating. In general, the culture format often depends on where the cells originally came from in the body. For example, HeLa cells came from cervical cancer and HEK293 came from fetal kidney. These tissues are made of cells that are usually attached to each other. Meanwhile, suspension cells like HL60 and JAGAT came from blood. As you know, blood cells naturally float in the bloodstream. Sometimes we grow more than one type of cell together. This is called co-culture. A good example is growing iPS cells on the top of feeder cells. And when I was a student at the university, I dealt with microglia, which is the one of brain cells. And I cultivated microglia with astrocyte. Astrocyte are attached to the bottom of cell culture vessel, and the microglia are floating in the cell culture media. So this is the one of example of co-culture. And also sometimes we grow more than Also, cell can sometimes grow in groups called the colony, or they can form round 3D shapes like a spheroid. This is the colony, and this is the example of spheroid, neurospheroid or embryoid bodies. As you can see, there are many different ways to culture cells. There are many types of containers used for cell culture. So let's talk about container. The most common ones are culture dishes and culture flasks. These come in different sizes, so please choose the right one depending on your culture scale. For flasks like this, we often use names like T25 or T75. The number shows the surface area where cells can grow. Flasks also have two types of caps, a normal cap and a filter cap. In case of normal cap, you need to loosen slightly to allow air exchanges gases. Meanwhile, for filter cap, we don't need to loosen them. About mulch well plate here, most have a flat bottom, but there are also types with a round bottom or V-shaped bottom. And for experiments using fluorescence or luminescence, some plates have clear bottoms and black or white walls. These colors help improve signal detection. Spinner flasks here is used to grow suspension cells in large scale. It has a stalker that keeps the cell moving. A chamber slide here is useful when using the microscopy, especially in tests like immunocytochemistry. There are many types of cell culture vessels. Please choose the one that best fits your cell type, culture size, and experiment purpose. As generally, there are various Belson medium such as DMAM, LPMI, or HAMS F12 or something like that. And medium are used to depend on your cells. In most cases, when you buy the cells at the cell provider, as I explained, you can also get the information to maintain the cells. Or another case, we refer to previous paper. This is for your information. This is the explanation of each Belson model. The meme is really famous now. The meme was modified by Dalvik based on meme, so-called the meme. He won the Nobel Prize for oncovirus, not cell culture media. The meme is widely used for adherent cells. LPMI is also famous. LPMI was developed at Roswell Park Memorial Institute, LPMI. As improvements were made, it was changed to 1629, 1630, and 1634. But I've never seen other LPMI commercial available. I just see LPMI 1640, often used for culturing floating or suspension cells. And about the DMEM HAMS F12, this is one mixture of DMEM and HAMS F12, used for fibroblast culture, and also the many medium for iPS cells are made based on DMEM HAM F12. Medium contains amino acid, vitamin, fatty, mineral, and glucose. There are a lot of different types within this medium. For example, about the meme, we or other companies such as Giveco or Sigmund have several types DMEM. We provide 12 types DMEM actually. It's complicated, so I will share key points of the components of basal medium. At first, glucose. Almost the case, there are three types, high, low, and without. You can choose them depending on your experiments. About glutamine. With and without glutamine. Glutamine is easy to break down. That's why some researchers prepare the medium without glutamine and add them before use. Or another case, you can use stable glutamine, L-alanine, L-glutamine, or Glutamax, which is the Samos brand name. And about HEPAS. HEPAS has more buffering capacity than sodium bicarbonate and used HEPAS with sodium carbonate. Sodium bicarbonate are buffering commonly used for maintaining the pH of cell culture. You use sodium bicarbonate in presence of 5% carbon dioxide, CO2. CO2 in incubator and sodium bicarbonate in the medium adjust the pH of medium to proper pH. So outside the incubator, pH buffering capacity is weakened. HEPAS has more buffering capacity than sodium bicarbonate. That's why HEPAS containing is preferred if the work is often done outside the incubator. But please remember that sodium bicarbonate is nutritionally necessary for most cells. So HEPAS should be added in addition to, not in place of, sodium bicarbonate. And also, HEPAS might show toxicity. Next, about phenol red. Okay, phenol red is a pH indicator. If the color of medium with phenol red is purple red, the pH is alkaline. On the other hand, yellow, the pH is acid. We can know cell condition by the color of medium with phenol red. But phenol red interfere to absorbance measurement for cell-based assay like MTT assay because of their color. And moreover, phenol red pretend as estrogen, which is one of hormone. So, if your cells are sensitive for estrogen, such as MCF7, which is a human breast cancer, you should avoid using phenol red. And the final point, sodium pyruvate. A little complicated. Cell provide ATP here for the energy for living. And to produce this ATP, they have to turn over TCA cycle based on pyruvate. Usually, cells make pyruvate from glucose via glycolytic pathway. But please imagine that when you are very tired, it is better to take sugar directly than rice for immediate energy, right? Of course, this is a kind of metaphor, but like this, pyruvate is often used for fast-growing cells or in specific conditions, such as low season culture or cloning experiments. So like this, there are many different types of demand with different composition. So please be careful when you order based on media. This is all second topics. Sorry. And we move to next topics. Aseptic techniques. And there are two types, cross-contamination and contamination with microorganisms. About the cross-contamination, cross-contamination is that your cells are contaminated with another cell unintentionally. For example, you originally culture HeLa cell, which is the adherent cells, but there are a lot of floating cell, suspension cell. In this case, you can doubt cross-contamination. To prevent cross-contamination, you never have to deal with more than 2 type cells at the same time. During waiting for centrifuge, you may want to passage another cell, but we do not recommend it. About the contamination with microorganisms, this picture are the example of contaminations. Do you know where contaminations are? With bacteria here, a very small one outside the cells. With fungus, this is largest size compared with cell, and it has branches like this. If contaminations are occurred in your cells, unfortunately, you should give them up. Of course, there are some ways to rescue cells which are contaminated, but it is hard and difficult work actually. I introduced general bacteria, but there is another difficult microorganism, that is mycoplasma. Let me share about mycoplasma shortly. Mycoplasma is one of bacteria, and in terms of their size is smaller than general bacteria. That's why it is difficult to detect them with ordinary microscopy and remove them by 0.2 micron filtration. And this point is most severe problem about mycoplasma. When the contamination of mycoplasma occurs, the appearance of cells and the color of cell culture media are not changed. That means that you can't notice the contamination of mycoplasma, but the data like cell toxicity test or functional test are weird. And then notice, oh, that is contaminated. Of course, it depends on the institution. Some researchers check mycoplasma in their cell regularly. Here are the detection method for mycoplasma. DNA staining is famous in Japan because it is recognized in the Japanese pharmacopoeia. Cells are stained by HEKIST or DAPI and compared with control cells. Can you see small pieces around the cells on this picture? They are mycoplasmin. Enzymatic detection are recognized in USB. Microalert produced by London are famous for detection kit. And about the microstrip here, based on isothermal PCR, it is really easy to detect them. Apical sell these items, so if you are interested in, please let Apical staff Tashir to know. And to prevent contaminations with microorganisms, antibiotics are added. That I would say this might be the answer for the question. Here are famous antibiotics for cell culture. Each antibiotic has its own target. For example, penicillin G targets bacteria, mainly bacteria gram-positive. Meanwhile, streptomycin targets both gram-positive and negative. That's why they are used together. And also, if you want to prevent the contamination of fungi or yeast, amphotericin B is useful. In terms of mycoplasma, gentamicin and or kanamycin are used. But please note that some researchers hesitate to use them because antibiotic-resistant bacteria may occur. Indeed, I usually deal with cell line without antibiotics, but one of my colleagues uses them because my colleague deals with the primary cells extracted from mouse brain. In the process of extracting cells from brain, it is easy to be contaminated. And also the loss of liver and time when contamination occurs is greater in primary cells than in cell line. So please consider the risk and benefit of using it. And these are our products for antibiotics to prevent the contamination. We have several products for antibiotics, and regarding penicillin, streptomycin, and amphotericin B, we have 2 or 3 mixture of them. This product is a 3 mixture, and this is a 2 mixture. And also this product here. This product includes glutamine because many researchers add penicillin, streptomycin, and also glutamine to their cell culture media before use. And here are for mycoplasma, gentamicin, and kanamycin. We offer liquid adjusted forms as well as powder forms. Of course, as supplier, we would like you to use a lot of antibiotics. But if it is possible to avoid using antibiotics, so much the better. So next, let's review aseptic techniques. Excuse me. Let's review the basic operations for using a clean bench. First, preparation. Before using the bench, turn on the UV lights and clean the working area. Be aware that UV lights is harmful to humans and the cells, so make sure to turn off the light before starting your work. Then run the circulating fan for about 5 minutes in advance. Next, check your cells. Key checkpoints are dramatic color changes in the medium, contamination of foreign materials, and also abnormal shape or proliferation rate of cells. If you notice any of these issues, you should discard the affected sample. Spray ethanol and clean working area. Also, instrument and medium should be wiped by ethanol. Next, aseptic techniques itself. Control your working area in the clean bench. Keep the clean bench free of unnecessary items and bring only the essentials into the bench. Additionally, as the airflow in front of a clean bench is easily disrupted, use the center of the bench. During operation, pay close attention to the pipette tip and the mouth of the culture vessel to prevent contamination. Reagent bottles and the culture vessels should only have their lid open during operation. And the operator's hand should not pass over container with open lid. When placing dish or tube lids, either orientation of the lid upside and downside is acceptable. If you spill culture media over reagents, wipe it up immediately with alcohol. Leaving the spill as it can lead to bacteria contamination. Do not talk to operators during aseptic operations. It is important to prepare the environment and physical condition so that you can concentrate on the operation. Last, cleaning. Bring instruments and reagent outside the clean bench. They may be contaminated by droplet on invisible culture media, so wipe them with alcohol to prevent carryover for next use. Wipe the working area, surface, and both sides of the clean bench's front glass with alcohol. Run the circulation fan for at least 5 minutes after work, then close the clean bench door and turn off the fan. Turn on the UV lights. Since ultraviolet rays degrade plastic, it's best not to leave it on for too long. Turn it off after about 1 hour. This is all for aseptic techniques. This might be a bit obvious, but I hope it helps, even just a little, with your further research. Next topics, subculture, and also in this section, in these topics, I will talk about the cell detachment, including the trypsin. Okay, and the next topic, subculture. This is the workflow of subculture. This shows a typical workflow. Depending on your cells, the way may be different. So when you subculture a new cell, please check the dataset which cell provider offer. As culture times go on, cell numbers increase. During proliferation, cells take up nutrients in media like glucose, amino acid, or vitamin, and release metabolites. In the cell culture vessel, the nutrients are losing, the metabolites are filling. Finally, cells are dying because of much metabolites and the depletion of nutrients. Before this bad condition, cells have to be transferred to new cell culture vessel with fresh medium. I mean, perform passaging cells. Or another case, cells in good condition are preserved for backup of valuable cells or because of long vacation. These topics, this session, at first, I will share cell detachment and cell count involved with passaging cells. As I mentioned, there are two types of cells, suspension cells and adherent cells. Cell detachment are done for adherent cell. In case of suspension cell, cell detachment are not needed. This is our legend for cell detachment or dissociation. Various types with different trypsin and EDTA concentrations are available. So that's why sometimes some research is complicated, but please choose depend on your experiment. So this might be my answer for the question. If you feel the trypsin EDTA is not fully affected, at that time, as a one option, you might try other concentration of trypsin. I mean, the concentration of trypsin increased. And so this is the trypsin. And also about the Actis and the Acumex. This product is ready to use cell detachment or dissociation solution, which is non-mammalian, non-bacterial replacement for all applications of trypsin. Gentle and efficient dissociation can be achieved, and no neutralization steps by CDAM or trypsin inhibitors are required. So easy to use. Acumax contain the same enzymes as Actis, but is formulated at a concentration that is 3 times higher and does not contain phenolate. This is mainly used for dissociation for spheroid. And what is detachment? Cells are attached to cell culture vessels or each other via several factors like cell adhesion factor, which is the protein, and metal ion. And the detachment reagent work for these factors. For cell adherent factor, trypsin work, and for metal ion, EDTA works. So generally, trypsin EDTA solutions are used for detachment. And Actis and Acumax are the mixture of proteinase, collagenase, and EDTA. Let's talk about more practical things, how to detachment cells. Before detachment the cells, please observe the cells and check the cell density. 70, 80, or 90% of confluent is okay, but 100% of confluent is bad. The color of medium changed to yellow because of metabolites. The shape here of cells were distorted and part of cells were floating. Before this bad condition, please perform passaging cells or preserve cells. I'll show you the practical operation of cell detachment. At first, remove the medium using an aspirator. And the cells are washed by PBS. Add PBS gently along the wall of cell culture vessel, like this. and then remove the PBS. Next, add trypsin EDTA. Incubate the cell culture vessel at the After that, incubate the cell culture vessel at 37 degrees at 5% CO2 in an incubator. During incubation, cells change to round and get to float gradually. Can you see? Cells change to round gradually. The incubation time is about from 3 to 5 minutes. If needed, you can prolong the incubation time to 10 minutes. But we do not recommend to incubate for 10 minutes more because cells are damaged by trypsin. After incubation, please check the cells by your naked eyes. The solution became muddy and thick. You can see several clumps of cells. Add a new medium contained FBS to deactivate trypsin because FBS contained trypsin inhibitors. This step is called neutralization step. Pipette gently to isolate the single cells from the clumped cells. This step is kind of important. Transfer the cell suspension to a new tube. Centrifuge the tube. After centrifuging process, remove the supernatant. And resuspend the cells with fresh medium. This is just for your information. Here is the typical amount of growth medium and detachment solution used. As I mentioned in the note, here, this table is based on the experimental experience of Nacalai test researchers. Of course, it is not very different from general usage. That there are also reports that the behavior of cells can change depending on the amount of medium. So please optimize the medium for each cell type. By the way, this paper is really interesting. If you have time, I encourage you to read it. Next, cell counting. Transfer small amount of cell suspension to a new microtube in a clean bench. If needed, dilute the sales suspension with DPBS. Add the trypan-blue solution and pipette gently. The volume of trypan-blue added is from 1 to 0.1 part of sales suspension. Inject 10 microliter of stained sales suspension into the gap between the counting chamber and the cover glass. Please observe morphology, then count cell number. Please cells within 5 minutes after mixing. Please count the cells within 5 minutes after mixing our cell suspension and trypan-blue, because even living cells will begin to be damaged by trypan-blue and take up the dye. Counting chamber has 4 sections: 1, 2, 3, and 4. And this is the image of one section on counting chamber with stained cells. Count the cells in this, counting cells in this area, okay? White cells are living cells, and the black cells like this are dead cells. For cells located online, determine which side are to be counted in advance. For example, you may count the cells present on the upper and the right side lines, but not the bottom of left side. Count 4 sections and calculate the average of cell number. For example, in this case, I mixed 100 microliter of cell suspension and 100 liter of trypan-blue. That means cell suspension was diluted by 2 times. And the cell number I counted is 95, 90, 87, and 88. The average is 90. So from the dilution ratio here, 2 times, and the number of cell count average 90, we can note that the concentration of this cell suspension is 180 times 10 to 4 cells per ml. This is the operation of cell count with trypan-blue. And maybe that is half of today's session. So I would say we will have a short break. And after break, I answered several questions. And then we move to ice breaking, maybe fourth game. And then we move to next topics. Is it okay? We will have maybe 10 minutes break and until, 12 minutes, so until 11 a.m.
Okay, participants, we will be having a short break until 11 a.m. So you can have a short break and then come back at 11 a.m. Okay, it's 11 a.m. already. I think everyone is here. So, Miss Mana, I think you can continue the session. Miss Mana, I think your microphone is mute.
Excuse me.
Okay.
Okay, welcome back to our webinar, and we will resume from answering the question. There are many question. I'm very so glad to see
Yes,
that.
there are 4 questions, I think.
Yes. And about first one, yes.
6, 7, or 6.
If cells are left, in the incubator for several days, oh my gosh, and they become overconfluent with some detaching, can the remaining attached cells still be used, or they already considered unhealthy? That is a good question, and I understand the situation. But of course, this is depend on your situation. For example, if I were you, and after that, I would like to conduct the drug treatment and then test the cell toxicity. In this case, and then also I can use other fluiding vial for this cell line. I will recover the cells again, because the result of cytotoxicity test is not reputable. But of course, sometimes we deal with real cells. So in this case, I will conduct passaging cells and watch observe the proliferation rate. And if the proliferation rate is the same as usual, I will use it. Yeah. So that is a dependent situation. And the next question, is it okay to use cell scraper if the cells are not fully detached? Yes, of course, you can use cell scraper, but please note some cells are sensitive for physical treatment. So yeah, this is depend on your cells. Sometimes I use the cell scraper. And the next question. Sometimes the cell don't detach even after 5 minutes in trypsin EDTA. It is not okay to leave them for more than 10 minutes. What the best thing to do instead? That is a good question. If I were you, I wait until 10 minutes. And then, I touch the wall of the cell culture vessel or pipette them. I mean, by physical treatment, I detached. But of course, as I explained at the last question, some cells are sensitive for physical treatment. That's why actually that is a depend on cells. And sometimes I do that. And also recently I deal with Hep D2, and Hep D2 can't be detached using after the treatment of trypsin EDTA. So at that time I changed from trypsin EDTA to Actis. As I explained, Actis is very gentle and low toxicity for cell lines. That's why I prolonged the incubation time with Actis for 1 hour or something like that. And at that time, it works. So it's one of the options to change detachment solution. And the next question, sometimes some cells had hard times to attach after revival from liquid nitrogen. Yeah. May you suggest a reason? And it is okay if you put extra FBS during the revival just to improve the survival and attachment rate. Okay, yeah, that is a good question. And about the reason, maybe unfortunately many cells were died. That's why they can't, they couldn't attach at the bottom of cell culture vessel. And to increase the survival ratio during revival step, additional FVS added. Yeah, that is a good option. You know, during revival step, I mean, right now after revival, cells are very sensitive for environment. That's why this is one of good option. I mean, the nutrients during revival step are increased. That is a good option. And also sometimes other protection solution additive add. For example, IPF cells, we added activin B. So like this, you know, this is a good way to add or increase nutrients during the revival step, because the revival step is very sensitive for cells. And next question. I am confused about the cell count. We put the mixture of cells and the triple blue at the two parts of hematocyte matter, upper and below. So for the calculation, why just count 4 parts, not 8? I'm so sorry that you are more precise to count the cells. Yeah, of course, hematocyte matter has a 2 part, upper and bottom, so we can count 8 part. However, it's hard to count 8 part within 10 minutes, within several minutes. So I usually count just 4 part. But of course, 8 part is okay, no problem. And are there any differences in terms of viability of cells when cryopreserved in complete medium plus DMSO versus in serum plus DMSO only? Actually, next topics I will talk about the cryopreservation, freezing the cells. So I will talk about this. Okay. Oh, thank you. So we resume the presentation and we move to freezing cells. Okay. Brief explanation for cell freezing. First, cryogenic bio. You need 2 special containers for cell freezing. Cell will be stored for a long time at low temperature, such as minus 196 degrees. So, cryogenic tube must be highly airtight and resistant to low temperatures. That's why do not use normal microtube for cell preservation. There are several options for cryogenic tubes, such as external or internal cap. Here, this is the external and this is the internal cap. And also round bottom or self-standing. Please choose them according to your preference or experiment. Next, about the method and the cryopreservation solution. There are 2 famous methods, slow freezing method and the vitrification method. Freezing is harsh for cells, and the key factor of cell damage are 2: ice crystal formation, and the osmotic pressure difference. Okay, to avoid these 2 factors, these methods were discovered. Slow fleeting method is more popular than vitrification method and used for a wide range of cells. By fleeting slowly, ice crystal formulation is minimized as much as possible. Conventional preservation solution is conditional medium with 10% serum such as FBS. This FBS protects the cell damage during fleeting. And sometimes we add DMSO to this solution additionally. This might be answer for the question. Thinking about osmotic pressure difference, DMSO is effective to prevent the cell damage, but DMSO shows cell toxicity. Whether to avoid the damage from osmotic pressure differences or the cytotoxicity of the DMSO itself depend on the type of cells. So please choose with or without DMSO according to your cells. It is one good way to differ the dataset cell provider offer. Vitrification method is mainly used for ES or IPS cells. By fleeting rapidly using special cryopreservation solution such as DAP123. This method avoids ice crystal formulation. Today, I will explain about the slow fleeting method using our product Cell Readable 1. What is Cell Readable 1? Let me introduce our product in brief. Our Cell Readable 1 doesn't contain FBS. Instead of FBS, salicin, which is isolated from silkworm cocoon like this, and this salicin protects the cells from fleeting damage. Cell Readable 1 is ready to use and shows high cell recovery and viability. We have two types, with DMSO and without DMSO. Our product can be applicable to ES cells. Cell Readable 1 is used for slow fleeting method. For beautification, we also offer Cell Readable 1 Beautify. If you are interested in Cell Readable 1 Beautify, please let the test Cassia know later. And this is for your information. We check the cell viability for those cell type here. And also, while we don't have detailed result, this shows the usage history of Cell Readable 1. Okay, so we move to more practical thing. Let's explain about the slow fleeting method with Cell Readable 1. But for conventional method, the procedure is almost the same, okay? First, at the day before fleeting, verify that confluence is around 50 to 70% by microscopy, and then repress the medium. It is important to ensure that the cells are in the log phase of growth. Host stem cells are good for freezing. Collect cells using the standard procedure and count the cell number. Recording the cell number at the time of freezing is useful for checking seeding density and cell viability after thawing. And then centrifuge the tube and remove the supernatant. Resuspend the cell with Cell Readable 1. The proper cell density is at between 5 times 10 to 5th power and 1 times 10 to 7th power cells per ml. This is for conventional preservation medium too. Cryogenic vials should be leveled in advance. Dispense the cell suspension into cryogenic vials. Immediately store these tubes at -80 degrees. To prevent cell damage, please perform rapidly from resuspended cell with cryopreservation medium solution to freezing. Use a program freezer such as Bicell if necessary. Next, I explain cell recovery. At first, prepare suitable fresh medium for your cells in a tube for dilution of defrosted cell suspension. Remove the cryopreservation tube from the freezer and immediately place in a 37-degree water bath. To avoid contamination, make sure not to submerge the lid in water. Place the tube in water bath until being thawed, which takes about 1 or 2 minutes. To thaw it as quickly as possible, we do not recommend the thawing by leaving it at room temperature on the lab bench. And also, please use a warm bath at around 37 degrees instead of water at room temperature. Transfer the tube into a clean bench immediately. Wipe the tube with ethanol. And then transfer the cell suspension to cell culture medium prepared in advance. To prevent the cell toxicity from cryopreservation solution, please dilute cells with cell culture medium as soon as possible. As a general guideline, it is recommended to dilute it about 10-fold. Centrifuge the tube. And remove the supernatant. Resuspend the cells with fresh medium. And transfer them to culture vessel. And start it to cultivation. Okay, so this is all for these topics. And also we have one question in the chat box. Thank you. I am working with 3 different cell lines, each requiring a different culture medium, okay? For certain assays that require phenol red-free conditions, what medium option would be most suitable across these cell lines? Okay. Different cell lines each require different culture medium. Certain assays require phenol red-free condition. What medium options would be most suitable across? That means uh about the medium commonly used for these three cells, but these three cells require different culture medium. Of course, you can culture these cells under a phenol red-free condition, but be careful. We can't notice the contamination from the color change of medium. That's why we have to observe carefully to the morphology of cells itself. But of course, we don't need the phenol red. So you can use the medium without phenol red for not only assay but also maintaining. So does it make sense? Is it? My answer is good.
No, Ardila.
Hello.
Huh.
Okay.
Does Miss
Okay.
Mana answer your questions?
Okay. Okay. Basically, the question about my question is that I'm working on the before this for the RSSA and QSSA, and it seems like when I run the experiment using the the media that contain the phenol rate, it somehow interfere with the readings. So when I did checking with a previous paper, they use a phenol rate free medium. So what I do want to ask is that okay, normally when I grow the cells, I will just grow in the phenol rate medium, just normal, and then after that, when I do the seedings, I still in the phenol rate medium, and then when I do the treatment, that is when I noticed that some of the paper they usually divert into the phenol rate free medium. So I'm asking, is there any possible media that can replace this, because I want to avoid the interference of the phenol. Is that clear? Sorry.
Oh no. Let me confirm. Excuse me. Okay. Let me confirm. So the problem is, the issue is you have to use without phenol red media to assay something, right?
Yes.
And is it possible to replace phenol red without media for maintaining or no?
Not for maintaining, but for DAC. Only just for DAC. Meaning that usually if the reagent requires us to dissolve the reagent in the media, but I don't want it in the phenolate content media. I want it a phenolate free media. So is there any media option that available for this, or I can just use the PBS to replace?
Okay. So sorry. I would say that it's a kind of specific thing, and also, if needed, could you send me the message about the details, and I will check it, and I will just reply you. Is it okay?
Yeah, sure, can.
Okay, thank you.
Thank you.
Oh, thank you for asking. Okay, so we move to next topics, final topics. Okay, so we already know the basic flow of subcultures. Let me brief summarize. As sales increase at the proper timing, you need to either passage them or plead up them by freezing. We have already looked at how to subculture, how to count cells using triple Buloh, and how to freeze and recover them. In this way, you can expand your cells and maintain them. But for what purpose? Of course, to use the cells for experiments and to study various biological responses. In this session, in these topics, we will focus on application of cell culture, especially cell toxicity assay. Let's talk about the cytotoxicity assay. But before talking, I will share application related with cell culture. In cell-based analysis, we usually detect three main things: cell condition, protein, and nucleic acid, roughly speaking. Cell condition: cells are fine or not fine. Cells are died or not died. If the cell died, how they die? Famous examples are cell toxicity tests such as MTT, WSD8, and LDH assay, or apoptosis assay using Annex 5. Next, protein. What protein increased or not? How protein moved? The famous example are Western blotting, ELISA, and immunocytochemistry. Lastly, nucleic acid. Which gene worked? The famous one is PCR, included real-time PCR or RT-PCR. Today, we will focus on cell condition. To check cell condition, there are mainly two types of tests: live dead assay or cell count, and apoptosis assay. First we talking about the live dead assay, cell count assay. We check if cells are alive or dead by looking at certain indicators. For example, to find living cells, we can measure ATP levels and also ATP levels, like using luciferase-based assay, or checking enzyme activity such as esterase and dehydrogenase. The MTT assay, which I will explain later, check for NADH, a type of dehydrogenase. To detect damage or dead cells, we look for membrane damage by the uptake or leakage of cell contents. As you know, apoptosis is a kind of programmed cell death. It includes special events like caspase activation, changes in the cell membrane, and DNA fragmentation. By detecting these events, we can determine whether the cell is undergoing apoptosis and identify which stage of the process is it in. For next slide, I will explain live dead assay or cytotoxicity assay, the microplate-based colorimetric assay. I will share the microplate-based calorimetric assay used for checking cell toxicity. The famous assay are MTT and LDH assay. Here is the mechanism of MTT, WSD8, and LDH assay. MTT and the WSD8, like this, detects living cells. On the other hand, LDH here detects dead cells. And these are based on oxidation reduction reaction. At first, I introduced MTT and WSD8. MTT assay is famous for detection of living cells, and WSD8 assay is similar to MTT assay. Please remember that WSD8 assay are advanced assay of MTT easier. Both of them contain tetradoyl salt, and LDH, which living cells have, reduced this tetradoyl salt, resulting in generate hormone dye. The difference between MTT and WSD8 is a type of tetradoyl salt. Actually, MTT and WSD8 are the name of tetradoyl salt. Hormone dye produced by WSD8 and NADH is water-soluble, but MTT is not. You can skip the dissolving step of formaldehyde dye when choosing WSD8. And to be honest, this dissolving step required with MTT assay is so hard work. Simply, WSD8 is easier than MTT. So once you start using WSD8, you could not come back to MTT. Next, about LDH assay. When cells got damaged resulting in cell wall were broken partially, internal LDH goes from cell to medium. LDH assay is to detect LDH level in cell culture medium. Here is protocol for MTT assay. Protocol is not complicated. Add MTT solution to the cells you are testing and then put it in a cell incubator. During incubation, purple crystal, purple participate, which is a formaldehyde dye, are generated. After incubation, solve the dye by adding solidization solution. This step is really tough work. You have to pipette one by one, or after sealing, let the plate at 37 degrees overnight. After dissolving completely, measure the absorbance at 570 nanometer. And here is the protocol for the WSD8. If you are used to MTT assay, you will definitely be surprised by how easy this method is to handle. Simple, add 10 microliter of the reagent and then incubate the plate. The incubation time is usually between 1 to 4 hours. During incubation, during this incubation, formaldehyde dye is generated. But unlike the dye produced in the MTT assay, the formaldehyde from WSD8 is water-soluble. This means you don't need to dissolve the dye with a pipette or leave it overnight. So after the incubation, you can measure the absorbance at 450 nanometer directly. I would like to share several point tips for this assay. These tips are useful not only for our MTT and WSD8 assay kit, but also for general MTT assays, okay? First, control. in MTT assay and WSD8 assay, setting the right control is very important. You need to use proper controls. Since it's a bit complicated, I will explain it step by step. First, the sample here. First, the sample you want to test. For example, a sample treated with drugs. Then, the negative control. This is a sample that is not treated with any drugs and show 100% cell viability. And next, we have the background sample. This is used to measure the absorbance of the culture medium itself. We subtract this value from the test sample and the negative control to collect them. Here, how it works. When you subtract the background from the test sample, so that means here, you get the number of living cells after drug treatment. And when you subtract the background from the negative control, you get the value of 100% cell survival. By comparing these two values, you can calculate the cell survival rate ratio after drug treatment. Here is a slightly more complicated case. This happens when the drug has reducing properties or already has its own color, okay? Because as I explained, MTT and also WSD8 are based on oxidation reduction reaction. That's why if the drug has reducing properties itself, it interferes this assay. And also about the color, WSD8 and the MTT is based on colorimetric method. That's why if the drug had the color, also this interferes the assay itself. Okay. So in such cases, you need to correct for this by adding the drug to the background sample here. For example, if you want to make a survival curve using a drug like this, and the drug has reducing effects or is colored, you must prepare background samples with the drug added at each concentration, okay? In this case, you will need a total 7 background samples. 6 background samples with the drug added at a concentration of 0.5%, 0.15%, 0.05%, 0.05%, 0.5%, 0.15%, 0.05%, 0.015%, and also 0.005% and 0.0015%. I mean that you need a sample at each concentration and 1 background sample without the drug for collecting the negative control. And next, optimization. NADH, which are target enzyme to detect in MTT WSD8, vary depend on the cell type. This graph shows how cell type affect the cell number and the color development time in the MTT assay. For example, HeLa cells here show an absorbance of 0.5 when 5,000 cells are seeded, while HL60 cells need 20,000 cells to reach the same level. This is because the amount of NADH differs depending on the cell type, which affects how much color develops. The same trend is seen with WSD8. Different cell type give different color intensities. That's why we need to optimize the cell number and also color development time for fast use or when changing cell type and medium. In this graph, cell number here means the number of cells at the time of seeding. The cells are incubated overnight and measured the next day. Since cells grow after seeding, it's important to follow the same time schedule as your actual experiment when doing your cell number test. Cell growth can vary with different media, so please also perform optimization when changing medium. Additionally, for MTT assays, it's important to test color development time. Once you stop the MTT reaction and measure absorbance, you can't measure again if the color isn't enough. But with WSD8, you can measure, then keep reacting longer if needed, and measure again. So WSD8 doesn't need such strict testing for color development time. Here's a quick explanation about the cell number optimization. Let's say you want to test the toxicity drug A here on hair cells after 24 hours. In the actual experiment, you seed the cells on day 1, incubate overnight, and drug A the next morning, add drug A the next morning, day 2. Then incubate for another 24 hours before doing assay. For optimization here, seed the cells 40 hours before the assay. I mean the 60 hours pre-incubation plus 24 hours drug treatment. Use a two-fold dilution series like this for cell seeding. If you are doing an MTT assay, prepare 3 plates to test the different color development times. This isn't needed for WSD8. After incubation, do the assay and get these results. And choose the cell number that shows a linear result here. And the absorbance under 2. This absorbance represents 100% cell survival in the main experiments. This is just my approach, but during optimization, I also prepare a background sample with drug A added at the highest planned concentration and check the result. If there is more than a 10% difference between the background sample without drug A and the one with drug A, then I can assume that drug A interferes measurement itself. As I mentioned earlier, I prepare background samples with drug A added at each concentration for the main experiments. And finally, about measuring at the reference wavelengths. To collect for turbidity caused by cells, we recommended measuring and collecting using a reference wavelength. With a microplate reader, if you check the reference wavelengths option and enter the wavelengths, the reader will automatically display the collected value. If your device doesn't have this function, you can measure both the main and the difference wavelengths manually. Then subtract the absorbance at the difference wavelengths from that at the main wavelengths for each well. This gives you the collected absorbance value. We especially recommended this for MTT assay. In MTT assays, turbidity can occur due to isopropyl alcohol in the solubilization solution and the protein in the FBS contained. In the FBS contained in the medium. The graph here shows the absorbance at 650 nanometer after adding MTT reagent and the solubilization solution to the MEM with 0%, 5%, and also 10% FPS. By the way, 650 nanometer is often used for reference to collect for turbidity. You can see that absorbance at 650 nanometer increase with higher FBS concentration. For MTT assays, be sure to measure at the difference wavelengths to collect for this turbidity. By the way, there are several types of solubilization solutions commonly used for MTT assay. Hydrochloric acid, SDS, DMF, and isopropyl alcohol are frequently used options. The composition also varies depending on the manufacturer of the kit. Regarding our kit, solubilization solution contained isopropyl alcohol and hydrochloric acid. Because during the development phase of our MTT assay kit, isopropyl alcohol was chosen because it showed high solubility. And let's compare the MTT assay and the WST8 assay. MTT produce insoluble purple crystals, while WST8 makes a water-soluble reddish-brown product. That means WST8 doesn't need a solubilization step, but MTT does. But of course, MTT assay has a good advantage point. This affected by reducing reagent and also less influenced by phenol red, since it turns yellow in acid in solubilization solution. And also, I like this point. Easy to see where reagent was added because the reagent is red. And most importantly, it's cheap, especially if you make it yourself. But the downside is it needs a solubilization step, and once that's done, you can't remeasure. WST8, on the other hand, has no solubilization step, develops color faster, about 1 hour, and you can remeasure if needed. But it's more sensitive to phenol red and the reducing agent. The red reagent makes it hard to tell if it's being added, and it's more expensive. So please choose one according to your situation and needs. And next, I will explain about LDH assay briefly. MTT assay and WST8 assay detect living cells. Meanwhile, LDH assay detects dead cells by checking the concentration of LDH, which are leaking from cells to medium. This is a very important point, so please remember that the LDH assay measures the amount of LDH in the culture medium, okay? There are two types of LDH assays, the homogeneous assay and the non-homogeneous assay. In a homogeneous assay, the reagent is added directly to the medium containing the cells. In contrast, a non-homogeneous assay involves transferring the culture supernatant to separate the plate. then adding the reagent and measuring LDH activities there. One advantage of the non-homogeneous method is that the remaining cells can still be used for other assays such as MTT or WST8 assay. So why do we combine the LDH assay with MTT or WST8 assay? Let me give you an example. Imagine you are testing the toxicity of drug A using an MTT or WST8 assay. You find that the number of living cells decreased as the concentration of drug A increased here like this. But at this point, we don't know are the cells actually dying or have they just stopping proliferating? To answer that, we include the LDH assay. If the cells are dying and the membranes are damaged, LDH will be released into the culture medium. So we will see an increase in LDH activities like this. But if the cells are just not growing or are metabolically inactive, but still alive, then LDH level stay low. This brings us to a key question. Can we use only the LDH assay to detect the toxicity of drug A? The answer is not completely. The LDH assay is great for detecting cell death, especially when there's membrane damages. But it won't detect the cells that are alive but not functioning properly. For example, if they have stopped dividing or their metabolism is suppressed. In other words, MTT assay, WST8 assay, and the LDH assay only tell part of the story. That's why we combine LDH assay with MTT or WST8 assays, which measure cell viability and metabolic activities. Together, these assays give us a clearer and more complete picture of the drug's effects. We are almost out of the time, so I conclude my presentation here. If you are interested in the LDH assay, please feel free to refer to the introduction manual.